Welcome back to STEMLens. It is of great joy that most of you have been able to understand concepts better by watching our videos, as we just recently concluded the series on the concept of living thing. Well, in the next series of videos, we shall be talking about the description of the representatives of major groups of prokaryotes and eukaryotes. That we shall be talking about major representatives of the different kingdoms we have. Alright, so for a start, we have Kingdom Prokaryote, that's the Kingdom Monera, but we shall be discussing that in the next video while we move to Kingdom Protoctista, because we are being referenced to another page for the Kingdom Bacteria, so I'll have to go to that page, and that would be in the next video. So in this video, we shall be talking about the Kingdom Proctoctista. First of all, what you want to know is that the Kingdom Proctoctista is also known as the Kingdom Protista. Representatives, we have the Chlamydomonas, we have the Amoeba, we have the Paramecium, Euglena, and Spirogyra. So if you missed our previous videos, the link is in the description, where you can get to know better about the Kingdom Protista, where we discussed about the characteristic features of the Kingdom Protista, as this video is just going to be about the major representatives, where I shall be discussing Chlamydomonas and Spirogyra. Now, the Chlamydomonas is oval or spherical in shape and is bounded by a cell wall consisting of hemicellulose rather than cellulose. Now, what is the difference between hemicellulose and cellulose? Because we are talking about the Chlamydomonas being bounded by a cell wall consisting of hemicellulose rather than cellulose. Now, they are both polyols, alright, they are polysaccharides. But now, hemicellulose consists of short branched polysaccharides, while cellulose consists of long unbranched polysaccharides. So that is the major difference. So Chlamydomonas is bounded by a cell wall consisting of long branched polysaccharide. Now at the anterior end, now when we talk about anterior, we have anterior and posterior. Anterior means close to the head or the forward, while posterior is close to the down or the caudal point or the bottom. Alright, so at the anterior end, on the right, you can see a picture that's a structure of Chlamydomonas rivalis. Right, so we'll come back to the diagram, but let us discuss the structure first of all. Now it says at the anterior end, the wall projects and thickens into a colorless papilla. Alright, so you can see the papilla here. You can see the papilla here. You can see the papilla here. So we have the papilla. It says two flagella which arise from the anterior end are used for movement. Right, we can see the flagellum. A single is called flagellum. The two are called flagella. Right, so two flagella arise from the anterior end. That's from the end of the head. Alright, now it says close to the base of the flagella are two contractile vacuoles which are used for osmoregulation. What is osmoregulation? What are contractile vacuoles? When we talk about cell, we shall talk about contractile vacuoles and all of that. But now osmoregulation has to do with the regulation of what? Osmotic fluids. Alright, so these contractile vacuoles most especially for the excretion of water, thereby regulating the amount of fluid in the body of the Chlamydomonas. Now, within the cytoplasm is the nucleus and a cup-shaped chloroplast which contains chlorophyll. Now, don't forget, in our previous video, we've talked and discussed on the Kingdom Protista being an eukaryotic kingdom. Alright, so here we have a nucleus that is bound by a nuclear membrane. So it is not just localized in the cytoplasm. Alright, so we have, okay, it says a pyranode not responsible for the storage of starch occurs within the chloroplast towards the posterior end, that's towards the bottom end. Alright, an eye spot is embedded on one side of the cell close to the cell at the anterior end. The eye spot responds to light stimulus so that the organism moves towards the source of light for photosynthesis. Alright, so it says Chlamydomonas is an organism that shares both the characteristics of plants and animals. The presence of flagella for locomotion, eye spots which respond to light stimulus, and contractile vacuoles for osmoregulation have compelled zoologists to claim it as an animal, while the presence of chloroplasts containing chlorophyll for photosynthesis and storage of excess carbohydrate as starch have made biologists to place Chlamydomonas into plant kingdom without dispute. So now, as discussed earlier, let us now look at the structure of this Chlamydomonas rivalis. So we can see, starting from the flagellum, which is for what? Locomotion. Then we can see the papilla, You can see the papilla. We can see the contractile vacuole for osmoregulation. We can see the eye spots, which serves as the spots for stimulus, receive stimulus, that's light, so that it can move towards the source to carry out photosynthesis. The photosynthesis requires the utilization of what? Sun energy, right? Now we have the cytoplasm. Cytoplasm is the general fluid that houses other organelles in a cell. Right? We have the nucleolus. In the nucleus, we have a cup-shaped chloroplast responsible for what? Photosynthesis as well. We have the pyrenoid, the site of starch formation and storage. Then we have the hemicellular cell, which I spoke about to be consisted of short branched polysaccharides. All right, so that is the first example. We shall be talking about the second example, which was Spirogyra. So these are two examples we shall be talking about. Now, Spirogyra is an unbranched. Take note, this question is a JAMB question. If you are writing JAMB, take note. It is an unbranched green filamentous algae consisting of a chain of cylindrical cells. Now, you may see questions like Spirogyra, whether it is branched, whether it is unbranched, the type of cell, if it is cylindrical cell, if it could be cylindrical cell, it could be cubic cells. They could just try to confuse you. Alright, so you must take note that it consists of unbranched green filamentous algae consisting of chain of cylindrical cells. Now, all the cells are similar. Each cell is cylindrical in shape with transverse end walls. That's the walls are transverse, opposite each other, lying beside each other. The cell is enclosed in a thick cellulose cell wall. The entire filament is covered with a layer of mucilage. Mucilage is like a gluey kind of layer, alright, which makes it slimy. Alright, makes it slimy. Inside each cell is a lining of ectoplasm, which encloses a large central vacuole. In the cytoplasm, the chloroplasts are arranged spirally. We'll look at the diagram. Each chloroplast contains pyrenoid. The nucleus is suspended by cytoplasmic thread. Take note, the nucleus is suspended by what? Cytoplasmic thread. Now let us look at the diagram of this Spirogyra. So here you can see the diagram of the Spirogyra by the left, figure 2.2. So we can see the cellulose cell wall. We can see the mucilage that's over the cellulose cell wall. We can see the chloroplast spirally arranged. We can see the pyrenoid. We can see the cytoplasmic strand that is what suspending the nucleus. Right, that's how to identify your nucleus. You may be confused sometimes, right, but you would always see some cytoplasmic strand around if it is a clear diagram. Then we can see the vacuole. Alright, so that's that. This kingdom is, should I say, the simplest, right? The simplest, although we are still going to talk about bacteria in the next video. So we have some other examples like Amoeba, Euglena, Spirogyra. Now, even this Euglena can be likened to the Chlamydomonas, because Euglena is also considered both plant and animal-like. Alright, we also have some other examples of such. We have Paramecium, Amoeba. So you can work on the Paramecium Amoeba, do your research on Paramecium Amoeba. They're also important, especially if you are going for WAEC. Alright, we have Paramecium and Amoeba. So that's all for this video. Thank you for watching. See you in the next video.